is enough to denature most proteins. Why don't the proteins in our kidney denature? And the reason why is because they're accompanied by protecting osmolytes. In the shark, it's accompanied by TMAO, which is poisonous for us, but we have glycine betaine. And those are counteracting protecting osmolytes that protect our proteins from denaturation as a consequence of urea. So there's a lot of work that's been done on the thermodynamics of osmolytes. And what we know is that protecting osmolytes shift this folding equilibrium towards the end state, and denaturing osmolytes shift the equilibrium towards the unfolded state. This one is protecting. These are all protecting. That's denaturing. Here's a review article with Wayne on this whole topic. But most of the work in this area has not been done by me. It's been done by Wayne or Tom Record or others. And I'll show you some records. How is it, thermodynamically speaking, that these osmolytes are doing? And what they know, and we're confident that we know this because it's straight thermodynamics, it's been measured. And it's been measured by a number of investigators. What we know thermodynamically, not mechanistically, is that the way they're doing it is that if you had a population of proteins under denaturing conditions, so here the unfolded state in water is lower in free energy than the native state, and you put in a protecting osmolyte, it raises the free energy of the native state, but it raises the free energy of the unfolded state much more, so that now the native state is lower in free energy than the unfolded state, and it folds the protein. Saying nothing about mechanism. And exactly the converse is true for denaturing osmolytes. It lowers the free energy of the native state, but it lowers the free energy of the unfolded state a whole lot more. So now the denaturing osmolyte can change a population in which the protein is native, the folded population dominates, the one in which the unfolded population dominates. Again, thermodynamically, what we know is that the predominant effect is on the unfolded state, and it's exerted predominantly on the backbone. Not on the folded state, and not on the side chain. So whatever osmolytes are doing, and they're doing it, and we know this thermodynamically, they're doing it to the backbone, not the side chain, and they're doing it on the unfolded state, not the folded state. And if they're doing it to the backbone, it makes perfect sense. Plausible that if what they're doing is predominantly to the backbone, it should be also predominantly on the unfolded state, because in the unfolded state there's more backbone exposed to solvent than in the folded state. This work was done initially by Chemika, and then more recently there's a paper by Matt Out and Wayne Gold. I should emphasize that in coming to these conclusions, which are sound thermodynamics, there were many, many complex problems that had to be solved along the way. And I don't have time to go into this. But doing this in a way that is thermodynamically sound turned out to be a real terrific thing.
Thank you, Owen, for your very kind introduction, and also for your strong support of the work that I am going to present while you were Scientific Director of NICHD. And in you, I also want to thank the NIH for supporting this basic research into molecular mechanisms of protein trafficking over the years. And I also want to thank the NIH for bestowing on me this honor of being able to present to you the work of my lab in what I consider my scientific home and in front of my scientific committee, which doesn't make it any easier. So this talk is going to be about sorting signals, adapters that recognize those signals, and the physiological processes in which they are involved. The talk will consist of two parts. First, I'm going to present a historical perspective on sorting signals and adapters to explain how we came about to study this problem. And in the second part, I'm going to present recent, still unpublished work showing a role for one particular adapter, the AP4 complex, in the sorting of the Alzheimer's disease amyloid precursor protein. As you know very well, the interior of eukaryotic cells is organized into a number of membrane-bound compartments, many of which you will be able to recognize in this electron micrograph of a pituitary cell. Now, each of these compartments has a distinct protein composition which is responsible for the properties of those organelles. So the problem that we and many other people in the field have been interested in studying is how is this heterogeneous distribution of proteins within cells achieved? In other words, how are some proteins targeted to some of these compartments, while other proteins are targeted to other compartments? In particular, we have focused on the organelles that make up the endosomal lysosomal system, which comprises various types of endosomes, such as sorting endosomes, recycling endosomes, late endosomes or multivesicular bodies, lysosomes, and a class of organelles that are known as lysosome-related organelles, or LROs, which includes melanosomes, platelet-dense bodies, cytotoxic granules, and others. These organelles are interconnected by vesicular transport pathways, and they are also connected to organelles that feed into endosomes and lysosomes, such as the trans-Golgi network and different domains of the plasma network. So there are some proteins then that are specifically localized to some of the organelles in this system. For instance, many of you are familiar with the transferrin receptor, which cycles between the plasma membrane sorting and recycling endosomes, but is not found in other organelles of this pathway, such as lysosomes. In contrast, there are other transmembrane proteins, such as LAMP1, which are mainly localized to lysosomes and late endosomes, but are not found in large quantities in the other organelles of the system. So the question, the fundamental question then in sorting in the endosomal lysosomal system is how are these proteins targeted to specific compartments within this pathway and within the cell. So already in the 1970s, it was proposed that transmembrane proteins likely had information that told them where to go within the cell. And it later, that information, it became clear that it was contained within the cytosolic domains of those transmembrane proteins in the form of an amino acid sequence. It was then proposed that those signals would be recognized by specific adapter molecules, which were components of protein coats, such as clathrin coats. So in essence, signal-adapter interactions led to the concentration of transmembrane proteins within coated areas of membranes of organelles. And a good example of that are clathrin-coated pits at the plasma membrane, which early on were recognized as the site for endocytosis of some receptors from the plasma membrane, as exemplified in this electron micrograph of LDL particles binding to their receptor and undergoing internalization into cells. Through the work of many people, including Ira Passan, who's here in the audience, these coated pits were shown to undergo a deeper invagination, eventually leading to the formation